Today's lesson will discuss the structure of proteins. Please refer to Chapter 18 in your textbook. Proteins have four types of structures: primary, secondary, tertiary, and quaternary. First, we'll begin looking at the primary structure. The primary structure is the simplest level of protein structure. It's the amino acid sequence that forms when amino acids bond together by peptide bonds. Each different protein has a different primary structure, and one small little change in that sequence of amino acids can affect the overall structure and function of a protein. The sequence of amino acids is determined in the DNA of the gene that encodes that protein, and this happens in the ribosomes of the cell. This diagram shows a protein's primary structure. Each amino acid is joined together by peptide bonds. Moving or changing this sequence will change the R group interactions. This, in turn, changes the protein structure. This is the case with a disease called sickle cell anemia. Sickle cell anemia is a disease that affects the shape of red blood cells. Red blood cells contain a protein called hemoglobin. Hemoglobin is made of 4 polypeptides. In 2 of these polypeptides, there is a very slight single change. The amino acid glutamic acid is replaced by valine. This one small change in the primary structure changes the shape of the red blood cells from a disc shape to a crescent shape. In this particular picture, it's showing you a portion of the amino acid chain. There's 300 amino acids in each of the 4 polypeptides that make up hemoglobin. And this is just showing a very small portion of one of the chains. It's showing where the amino acid difference is. And you can see in the top structure that we have glutamic acid, and because of a substitution of glutamic acid with valine, we go from our normal hemoglobin here to our sickle cell hemoglobin here with valine. And this is just one change out of 300 amino acids in that particular chain. Because of this amino acid substitution, the sickle cell hemoglobin molecules assemble or form into long fibers, causing this crescent shape. This causes the red blood cells to get stuck in blood vessels, and this inhibits proper blood flow. How does this affect a person with sickle cell? Well, it can cause shortness of breath, maybe dizzy spells, headaches, and maybe even pain in the abdomen due to the blood not being able to flow properly. This slide shows the difference between a regular disc-shaped red blood cell and a sickle cell. The sickle cells, you'll notice, are crescent-shaped red blood cells. I'm going to circle those in blue, and you can see we have areas of these more elongated or crescent-shaped red blood cells. Notice that not all of the cells are affected. The cells that are affected are the ones where the amino acid chain, that sequence didn't form correctly. And that's not going to always happen in every single sequence. So you'll notice here that we have some regular red blood cells as well. And the regular red blood cells are like this, these round ones, like so. Those are regular looking red blood cells. Secondary structure is where folding of the amino acid chain begins. This is due to interactions between atoms found in the amino acid backbone. Specifically, the type of bonding will be hydrogen bonding, and that will occur between the carbonyl group and the amino group. And I'm going going to show you how that occurs on the backbone. And if you remember, when we drew amino acids, we had this NCC backbone, and we had a carbonyl group, and we also had the amino group. Now, it's not necessarily going to be the carbonyl group and the amino group like I have drawn here right next to each other. Usually it's going to happen between it's going to happen like every fifth carbon and nitrogen. So it could be carbon 1 and then the nitrogen that would be after carbon 5. So it's not going to be right next to each other, but I want you to just kind of see why we have that hydrogen bonding occurring, and that's because we have this oxygen and this hydrogen in the backbone structure that's going to allow for that hydrogen bonding to occur. The hydrogen bonding does cause two types of structures to form. The one type is called the alpha helix, and the second type is called beta pleated sheets. In the alpha helix structure, like I said before, the hydrogen bonds form between the first carbonyl group and the fifth amino group, and this pattern actually pulls the polypeptide chain into a helical structure and sort of looks like a spring, like so. And each turn would have 3.6 amino acids. And what's going to happen when this forms is that the R groups are going to stick out off of the structure like so. Here's a better illustration. There's also illustrations in your book. But if you look closely at this model, you can see that we have oxygens here that are going to react with the hydrogens on the nitrogen group, and it's going to form that hydrogen bond. And you can see that all along the structure like so, that is occurring and pulling that structure into this alpha helix. Beta pleated sheets is the other form that the secondary structure may take, and this is where we have hydrogen bonding occurring, and it actually forms sheets almost like cardboard, and the R groups are found sticking up or down from these sheets. Let me show you what that might look like. And again, we're going to have our backbone like so, and then maybe we have another sheet here, and we have a carbonyl group and an amino group like so. And we would have some interactions occurring between the hydrogens and oxygens like so. This actually makes the protein not very stretchy, and it can stack a little easier because the major R group contributors are glycine, alanine, and serine. Whoops. And they're all very small R groups, so it allows for better stacking between the sheets, and so it allows it to not be very stretchy, whereas some other proteins, depending on what form they take, can be a little bit more stretchy. The beta pleated sheets can also take two forms. One would be parallel beta sheets, and the other would be anti-parallel beta sheets. And all that means is that in parallel sheets, the N-terminus ends line up together, and the C-terminus ends line up together, and you have hydrogen bonding occurring like so. And in anti-parallel sheets, you would have two polypeptides that would be folded on each other, where we have C-terminus and N-terminus lining up, and then N-terminus and C-terminus lining up on the other end, and then we have hydrogen bonds occurring like so. So it's just all in how the sheets are lining up, or the amino acid sequences are lining up. In parallel, we're lining up where they're the same, starting with C-terminus, ending with N-terminus, and anti-parallel, they would be opposites. And again, we'd have our R groups. I can show you better with this. Our R groups would be sticking straight up off of that alpha carbon like so, either above or below the structure. And it would look something like that if we were magnifying in. They'd be above or below the plane of that structure. The third structure we'll talk about is the tertiary structure. So we've talked about primary structure, where this is the amino acid sequence forming. It's more linear. And then we've talked about the secondary structure, where we've got hydrogen bonding occurring in that backbone of each amino acid, and it allows for hydrogen bonding to occur and either form an alpha helix or a beta pleated sheet. And after that occurs, the protein is going to start further folding in on itself, and this happens because of R group interactions. Now, we haven't seen any R group interactions in any of the other two structures, but here is where the R groups will start to interact, and there's 4 types of interactions. The first type is van der Waals, and if you remember anything about van der Waals, those are the London dispersion forces. So we can have London forces here. Sorry about that, London forces. And the other one is dipoles. So we can have dipole-dipole. And we've talked about those before when we were looking at interactions that happen between organic molecules. So again, we're looking at these atoms that are on these R groups, and some of them are going to be only held together by these weak van der Waals forces. Those would be our nonpolar groups. And those are the ones that are going to actually want to fold and be buried on the interior of a protein. And I'll show you pictures of what I'm talking about with that in a moment. The second type, again, we can have hydrogen bonding between our R groups, and hydrogen bonding would be happening between polar R groups. So van der Waals, I'll just put over to the side here. This would be nonpolar. Hydrogen bonding, again, would be polar. That's things that would have oxygens that can bond to other hydrogens. It could have nitrogens as well. Those would also be something that could hydrogen bond. These actually will like to stay on the outside of the protein because they like water, and the outside of the protein is going to be where it's in aqueous solution. The nonpolar molecules, they want to be buried on the interior because they don't like water. They're hydrophobic. The third type of bonding that can, or interaction that can occur, would be ionic bonds, or what we call salt bridges. These occur between positive and negative charges, like ionic compounds. Like we would have a positive charge and a negative charge that would be attracting. And if you remember, we have acidic and basic R groups, and they each have charges on them. So those would be the bonds that would be forming between the R groups that are acidic and basic. And the last type would be covalent bonding, And covalent bonds are between thiol groups. Thiol, if you remember, when we talked about thiols, that means we're talking about sulfur. So we call these disulfide bonds, and they join together almost like a safety pin. They're pretty strong as far as the types of interactions that occur between R groups, but they're only occurring between two sulfur groups. And I will again be showing you the disulfide bridges as well in a later slide. Now some proteins will stop here at the tertiary structure and they will be done forming into their protein. Once it gets to the tertiary structure, it's starting to define its biological function. And what's important with proteins, and we talked about this with sickle cell, that particular genetic disease, is because the protein chain is not formed correctly. So we go through each of these structures, and if something isn't formed correctly, that tertiary structure is going to end up not being able to function correctly. So the tertiary structure actually defines the biological function. And again, many proteins will only have 3 levels of structure. And some of those functions we have talked about before. They can be transport proteins, antibodies, regulatory proteins. They can be protein channels and cell membranes. They can be part of structure or movement in our bodies, parts of our muscles, our hair, our fingernails. So proteins make up a large portion of our bodies and have very specific functions based on their structure, which comes from the amino acid sequence that forms in the primary structure. I put this slide in here just to kind of give you a quick review of some of the R group interactions that maintain the tertiary structure or that formation of the protein. And so again, we have the covalent bonds or disulfide bridges between 2 cysteine residues. We have salt bridges. That would be ionic bonding between positive and negative charged portions, or our acidic or basic portions of R groups. We can have hydrogen bonding between polar residues. That would be our oxygens bonding with hydrogens. And we would also have hydrophobic interactions. Those would be our van der Waals, which include London forces and dipole-dipole interactions. This slide shows some of those R group interactions that I was talking about. In this particular slide, we have a protein that has started its tertiary folding, allowing for some of those R group interactions. And you can see here with the ionic bond, we have a positive amino group, we have a negative oxygen group. Again, we can be thinking acidic and basic groups. Those are going to be our polar interactions. Here we have hydrophobic interactions where we've got some nonpolar R groups. And you can notice that they're kind of buried in between this kind of loop here on the protein chain or amino acid chain. Here's our disulfide linkage where I've got the cysteine groups and the sulfurs are linking together, forming a pretty strong covalent bond, probably the strongest of all the R group interactions. And then we also have hydrogen bonds where you see we have an OH group on one R group and we have a carbonyl group on another, and they will form a hydrogen bond between the oxygen and the hydrogen of those 2 R groups. So that kind of gives you a little visual of how these R groups will interact and where they'll be located in a chain. Nonpolar interior, polar exterior. The other linkages are just going to continue to fold the protein into its proper shape and give it the function that it's going to need. The final structure we will talk about is the quaternary structure. This structure does not form with all proteins. It will actually be made up of more than one polypeptide, such as hemoglobin. That's the one of the examples of a quaternary structure. It's where more than one polypeptide comes together to form this particular protein. Like I said, some protein structures will stop at the tertiary structure, but in the case of hemoglobin, it's actually going to form the quaternary structure with 4 polypeptides coming together to form that. It's 4 subunits, and they're bound together by those same 4 interactions that we just got finished talking about with tertiary. These subunits are bound together with those same interactions, and this allows the protein then to become functional. One other thing that can be on quaternary structures or even tertiary structures would be something that we call a prosthetic group. And a prosthetic group is a non-protein that is bound to a protein. So one example of that, and probably the best example, is the heme group or the iron group that is on hemoglobin. That iron group on hemoglobin carries or holds the oxygen so it can be carried around in the bloodstream and moved where it needs to go out into the cells. And this figure here just shows you all 4 structures. The primary protein structure, where it's just the sequence of amino acids. The secondary structure, where it's starting to form alpha helix or beta pleated sheets, depending on what the protein is. The tertiary protein structure, where we've got the 3D folding with R group interactions, and then the quaternary structure, where we have more than one subunit or amino acid chain coming together to form the quaternary protein structure. The last thing I want to talk to you about is the difference between fibrous proteins that form and also globular proteins that form. And fibrous proteins would be proteins that are made usually from an alpha helix strand, and these strands come together, as you can see in the picture to the right of the slide. And these are our structural proteins. They're very strong. They have very strong mechanical strength. Structural proteins being things like fur, your hair, your fingernails, and they're arranged in fibers or sheets in the secondary structure. And most times, though, they are formed by the alpha helix coming together and just continuing to coil together with other alpha helixes to form these fibrous proteins. If beta pleated sheets are forming into the fibrous protein, one example of that would be silkworm produces what we call silk fibroin. It's made from anti-parallel sheets, and if you remember, that would be where we have the C terminus lining up with the N terminus, forming that anti-parallel sheet held together by hydrogen bonds. And these sheets come together, again, they have small R groups, and they can stack together almost like corrugated cardboard. If you know what corrugated cardboard looks like, it would be very similar to that look. These long sheets just sandwiched together to form that silk. Now, globular proteins are more compact. They have a more rounded shape, more of a 3D structure. They have more of our functional roles. They would be our antibodies, our enzymes, hormones, things like that would be the globular proteins. I promise this is the last slide of notes. I know this was a lot of information, but here's just a summary of the fibrous versus globular proteins. As far as the shape, its purpose, the amino acid sequence, what does that look like, its durability, examples of the types, and solubility in water. So just a real quick summary of those. You can look through that at your leisure. And the last slide is your assignments. Most of these assignments you probably have completed because they were assignments that were given while I was out sick last week, so you might have a majority of them completed, but I recommend that you look over sections 18.3 and 18 to 18.7 in your textbook just to reinforce these notes, and then answer the questions that you see on this slide. I know it seems like a lot, but again, I believe a lot of this you may already have completed and just have to submit it to the Dropbox. Please make sure that you submit to the appropriate Dropbox, and please make sure it's a handwritten assignment. You can submit a picture of your assignment if you wrote it in your notebook, or if you did it on Notability, you can send me a picture from there, or you can send it as a PDF, whichever works best for you, because I will accept either. So good luck with getting that assignment done and hand it in, and I'll be preparing our next lesson.